especially to Peter Morfise and to Peter Bandettini for providing the opportunity to present today via WebEx. I hope the audio and the video quality is sufficient so you can hear everything and see everything. My presentation is about the virtual brain simulation platform, and I'm going to focus particularly to how we can use the simulation framework in order to integrate multimodal data. So for those of you who have not heard about it, you can go to thevirtualbrain.org. The Virtual Brain is an open source software that is public since 2012. It has been downloaded since then more than 23,000 times. It's a software that is based on Python and that runs on different platforms, Windows, Mac OS, and Linux. And it can be deployed on your own notebook or personal computer and run there, but it also runs on high-performance computers. So at the heart of the virtual brain is the integration of computational modeling, theoretical neuroscience, and multimodal imaging. And I'm going to show you now, with the help of my colleague Jessica, whom you see here, how we construct virtual brains based on imaging data. And I just know that you are not seeing the video animation in the presenter view. I only see it on my big screen, unfortunately. So I'm going now to the next slide. That is actually a video that you cannot see, unfortunately. So I'm going to describe what you would see. This is again Jessica's head. This is an MRI image. We have multiple different types of MRI images available, and based on those we reconstruct different features of the brain of Jessica. So we reconstruct the different surfaces, the head surface, the cortical surface. We reconstruct the nerve fibers from the fusion tensor imaging data. We parcellate the brain in different brain regions. And then each brain region is represented by a population model, and the population models are interacting through the anatomical connectome. The brain then, or the brain model, simulates diverse types of activity, ranging from local field potentials, population spikes or firing rates, EEG, MEG, fMRI signals. And in order to produce the data that make out of a general brain network model, a personalized brain network model, we have published and generated several types of pipelines that we are continuously advancing. The latest version is now available publicly on several high-performance clusters, beneath others also on the collaboratory, the joint workspace or workbench of the European Human Brain Project. You might have heard about the European Human Brain Project that has a goal to build infrastructure for neuroscientists to share data, but also to share software and tools, and also computation resources and storage in order to make more benefit from the large amount of data that are available in the community. So the pipeline can be run via this joint workbench or workspace. It's deployed on the supercomputers that belong to the Human Brain Project, particularly the CSCS that is located in Switzerland. And everybody can get an account for this collaboratory and use the services and tools that are offered there. And it's also possible to apply for computational resources via the ECEI Phoenix program, and then use the resources from the supercomputing centers that are connected with HPP. The data that are processed by the pipeline are in bits format. So the pipelines are containerized applications. They are so-called bits apps, because they use data in the bits format, and they also generate derivative data in the bits format. We have also bits app that then converts the derivative data into TVB-compatible data sets that can be directly uploaded to the virtual brain and used to personalize our brain network models. So with a virtual brain, we are not simulating all individual 100 billion neurons in the brain. This is impossible, but we take benefit from a phenomenon that is ubiquitous in nature, which is synchronization. Here you see an example of starling murmurations. And here you see now a flock of neurons in the phase space spanned by the membrane potential and by the opening probability of the ion channels. And you see there's also synchronization, so they almost behave like a flock of birds. Oh, you don't see it because, sorry, it's not animated. I'm looking at my big screen. So you imagine now that you see the synchronization and imagine a flock of birds, a murmuration of starlings. And in order to describe this murmuration of starlings, we use a method from statistical physics, field approximation. You see in your still figure now the little red cross here, and this indicates a mean field approximation of this group of synchronizing neurons. So, as I said already, we are not simulating typically detailed networks of neurons, but population activity, as you can see here in the middle. Parameters from the cellular or microscopic level are averaged across the entire population. So for example, here, this population model that I'm showing here in the middle of the slide, you see the connectivity between the excitatory and the inhibitory mean field in terms of AMPA and NMDAergic coupling, and the feedback inhibition, the GABAergic coupling. These are parameters that are averaged across the population, and the values determine the dynamical repertoire of this population. And then each brain region is represented by such a population model. The virtual brain offers several kinds of population models. Currently, we have 15 different population models that can be selected depending on the research question that you are going to address. And these population models then are interacting through this anatomical skeleton and generating activity, local field potentials, firing rates, and then via forward models that are also part of the virtual brain package. We can compute MEG, EEG, and fMRI signals. So at the heart of the virtual brain is the evolution equation that you are seeing here. On the left-hand side, you see the differential operator. On the right-hand side, the vector of state variables. N stands for the population model that you have selected, and X for the state variables, which depend on the selection of the population model. And then the first sum operator shows how the state variables of all the connected nodes J are summed up, and taking into account the time delays delta or D in this equation that come from the structural connectivity matrix, from the distances between the region pairs, and also taking into account the connection weights that we also get from the fusion tensor imaging data that is in the structural connectivity matrix as C. Since these are relative values, we have also scaling factor G that is determined by fitting fitting the model to empirical data that could be, for example, functional connectivity or functional connectivity, dynamics, or also individual time series. It's also possible to run surface simulations. For this, we have the second sum operator and lateral connectivity kernel LC, which then is also scaled with the scaling factor L. One can also add a stimulus function I and also external noise. So in order to personalize now the model, we have different possibilities that are highlighted here. You can use the structural connectivity rates from an individual. You can use the structural connectivity distances from an individual. We also can use individual local connectivity, taking into account the surface geometry of the cortex. And we have the scaling factor G that is determined based on the individual functional data. So these are all more personal individual. So this was the classical approach. We are presently also developing multi-scale simulations, because not all features in empirical data can be described by coarse population-based brain network models. So for some questions we need more detail, and for this we are developing co-simulations where the majority of the regions of the brain are represented by population models, but some regions can be simulated in more detail at a microscopic scale. So in this example, we have the hippocampus, the entorhinal cortex, and the dopaminergic midbrain, represented by microscopically detailed networks and embedded in a network of population models. The anatomical connections for the detailed networks are taken in this case from the literature. Ideally, in the future, we plan to link available anatomical atlases that in part are still getting digitalized to feed this information directly into our models. So the benefit of such multi-scale models is that we can see how different are interacting. So there are causal influences between the large-scale model and the mesoscopic or microscopically detailed models. And in this example, we can see how, for example, neurodegeneration and the large-scale connectivity changes the processes at a microscopic scale that are relevant, for example, for memory consolidation. This co-simulation proof of concept is also available publicly in the Human Brain Project Collaboratory. There we have coupled TVB with NEST. NEST is a microscopically detailed simulator, very well advanced, with many different synaptic and spiking neuron models that one can select. And in this proof of concept, when the user can, in a Jupyter notebook that is made available, set up different types of connectivities or connections between the TVB simulator and the microscopically detailed networks that are simulated with NEST. And the simulation then is happening in parallel. So each time step is integrated in TVB and in NEST separately, and then the output is coupled. So this again is a video that you unfortunately cannot see. On the left side, you would see fluctuating activity that is simulated fMRI activity in the resting state of Jessica's brain. And beneath that, you would see the time course of the excitatory and the inhibitory population firing rate. So the advantage of integrating those empirical data in a computational model is that with this one can build hypotheses about what the underlying neuronal causes are. And here for each individual population model or brain region, we can recover the time courses of the firing rates of the excitatory and inhibitory populations. And this is something, of course, that we could not simply measure in Jessica's brain. On the right-hand side, you see the model that we are using here. This is a reduced Wong-Wang model. The topmost rows show the input currents for the excitatory and inhibitory population that are then translated into firing rates for the excitatory and inhibitory populations. This is what you could see in the video. The video is, by the way, also online together with the publication. If you are interested, you can watch it later. The firing rates then are translated into synaptic activities, again for the excitatory and for the inhibitory population. So with this approach, where we can reproduce macroscopic or mesoscopic observations, and by this we are generating a hypothesis or self-consistent theories how these observations could be generated. So here you see now in the upper panel features of the simulated data of a single model with a single fixed parameter set. And in the bottom panel, you see well-established features that have been published from empirical studies across different species and also across different timescales. On the left side, we have the faster timescale, on the right, the slower. And it's really exciting to see how a single brain network model with a single fixed parameter set can capture at the same time many different observations. So I'm starting now at panel A. There you see the relationship between the firing of neurons and the phase of the local field potentials. This is an alpha cycle. So in the previous talk by Mark Cohen, the alpha rhythm has been mentioned already, a prominent rhythm in humans, but also in monkeys. And in monkeys, this relationship has been explored systematically, and the specific relation between phase of alpha and firing has been established, and this relationship can be reproduced by the model. The ratio between inhibitory and excitatory input currents, the excitation-inhibition ratio, can be measured in rats and mice, has been published many times, and is reproduced by the model. The relationship, panel C now, between the firing rates and the alpha power is also reproduced by our model, has been systematically characterized in invasive monkey recordings. We also capture with the model the inverse relationship between the alpha rhythm power and the EEG and the both signal shown in panel D. We capture the scale-free behavior of resting state fMRI data, and we capture in panel F the switching of the functional connectivity. So many different features are encapsulated in a single model, and the nice thing is that by doing this, we can now see how changing the properties at one scale would also alter the properties of our observations at another scale. So we can manipulate and see what the consequences are, and we can also learn how the different snippets of observations are linked together mechanistically. And this is what we wanted to achieve. We want to turn the many different pieces of observations that we are having available into real understanding of the mechanisms of the brain. So another video that you can unfortunately not see, where in the left brain you see the EEG source alpha power, in the middle brain you see the simulations by the brain network model, and the right brain you see the resulting resting state fMRI activity, the simulated resting state activity. And this is the crucial and amazing part, that we are able to capture those resting state network fluctuations that we can also measure with our brain models. And with the help of our brain models, we can also reveal the underlying neuronal interactions that possibly give rise to these large-scale observations. So now the functional connectivity has been explored beneath others in the Human Connectome project, and there are some publications out that relate intelligence to functional connectivity. We have downloaded this data and found 742 of these individuals suitable for our virtual brain simulations. So we have virtualized their brains, and we could reproduce the relation between functional connectivity and intelligence. So on the left panel, you see the intelligence score versus the mean functional connectivity, and there's a significant relation between the two. On the right panel, you see the relation between mean functional connectivity and mean reaction time, and they are also related to each other. So in other words, higher intelligence is related to higher functional connectivity, and higher functional connectivity is related to higher reaction times. And higher intelligence is also related to longer reaction times. So in this intelligence task, those individuals who were scoring higher also needed longer response times to solve their tasks. So now that we are able to simulate functional connectivity, and functional connectivity obviously is related to cognitive function and in particular to intelligence, we can now try to use our brain network modeling approach in order to really understand the mechanisms behind and not just reporting the correlations. And this is what we are doing here. So we have built virtual brains for these Human Connectome Project individuals. And from these virtual brains, we can infer the input currents that drive the nodes, the populations. And here you see the average input currents driving the populations and the different virtual brains of the Human Connectome Project individuals related to functional connectivity. And what we found was that there's an inverse correlation between the functional connectivity and the input currents that drive the individual nodes of our virtual brains. So higher functional connectivity results in lower input currents. So now that we can construct these virtual brains for the individual for individuals with different intelligence scores, we can embed functional circuits that perform certain cognitive tasks in our brain network models and see how the different structural connectivity and the difference in the functional connectivity that is generated by those models affects the performance of those cognitive circuits. And we have done this. We use here circuits that can be used to simulate or mimic decision-making and working memory. You see this here on the right. These are two excitatory populations, A and B, that are mutually inhibiting via an inhibitory population. And the working memory can be simulated or mimicked by the circuit model, by persistent attractor states, and decision-making can be simulated by winner-take-it-all competition between the two excitatory populations A and B. So now we focus on the working memory performance. We see that stimulus in the upper panel, the little blue arrow, can elicit a persisting attractor state in one of the two excitatory populations. If the stimulus is too low, as you see in the right panel, then no persistent attractor state is formed. So the persistent attractor state mimics the memory. If we now apply 2 seconds later a second stimulus to the distractor population, shown in red, then either it is too weak and the persisting memory trace is not disturbed, and then the memory is robust, or the second stimulus disrupts this persistent memory trace. Or in the right panel, we have the situation where we have no memory trace from the start. So now we can look how this performs when we embed this in different models of individuals or groups with different intelligence. On the left, in the bottom panel, you see the low intelligence virtual brain, and on the right, you see the high intelligence virtual brain. For the high intelligence, we have seen previously, I showed that some slides ago, that the input currents, the average input currents that drive our regions are lower. For the low intelligence group, we have shown that the average input currents that drive our regions are higher. So the diagrams show now on the x-axis the current that goes to the distractor population, and on so on the y-axis, and on the x-axis we see the strengths of the mutual inhibition between the two excitatory populations. Blue shows the areas of memory disruption by the distracting stimulus. Red shows the areas where the memory trace is 100% robustly held. And white areas in the diagrams show the scenario where no memory trace has been generated at all. So now we can see in the bottom row on the left panel that already a small distracting stimulus is sufficient in order to disrupt the persistent working memory trace. If we look in contrast to the right panel for the high intelligence group, we see that larger stimuli are needed in order to disrupt the persistent memory trace. But we also see that we also need, in order to induce a memory trace from the start, we need higher stimuli. This means that stimuli with low intensity are considered as noise and will not lead to a persistent memory trace in the high intelligence group, which may have the benefit that in order to induce a memory trace, you need more salient stimuli. So noise is disregarded and the system is kept free in order to process more salient stimuli. So now for the decision-making circuit, we were inspired by a visual ambiguous motion of dots experiment, where unfortunately the video does not work again. So imagine that the dots on the right side are moving and there's a lot of noise, but there's a slightly stronger movement component to one of the two directions. This is mimicked by stimulating the excitatory populations A and B. One of the two populations gets slightly stronger inputs. And this is the contrast of the evidence. So if you have slightly more input, for example, in population A, we would have a positive contrast. This is now the result of our simulations. On the right side, for the low intelligence virtual brain, you see the firing rates of the two competing populations in blue and in red. The blue trace ramps up immediately after onset of the evidence accumulation that is indicated by the blue bar, and leads to a threshold that terminates a decision. On the left-hand side, for the high intelligence group, you see that the blue population ramps up much slower. So there's much more time for evidence accumulation in this system. And if you look now at the bottom panels and the middle, you see in the middle panel, on the x-axis, the input currents, and in blue, you see the evidence accumulation time. So we see that the lower the input currents in the background network, the higher is the integration time, the more time the system has for evidence accumulation. And if we look now in the right panel, we relate the input correlations, so basically that is the functional connectivity, to the performance of the system, independence of the chosen contrast. So the x-axis shows the contrast of the evidence, and the y-axis, and the x-axis shows the input correlation. The color in the right bottom panel indicates the precision of the decision. And we see that by increasing the input correlation in the system, we can increase the performance of the system. So white means there are 100% correct decisions, and black means that the performance is poor. If we now choose an intermediate contrast, let's say 2.6, just by changing the network input correlations, we can move the system from poor performance to excellent performance. So now these are interesting observations, but we want to really understand the laws and the rules that lead to these observations, and this is possible by using approaches from dynamical systems theory. So here you see the phase portrait in the lower row for 4 different network input correlations, ranging between 0 and 1 maximum correlation in the network. We also see the contrast that has been used for these simulations. C is 0.2, so there's a very low contrast between the 2 populations. The activity, the state variables of the 2 populations are the x and the y-axis. A positive contrast indicates that the A population, indicated by the x-axis, gets slightly more evidence. The red diagonal line is a separatrix that separates 2 attractor states that are indicated by these filled black circles. The gray or light purple lines in the background are the flow fields. The gray plots are the 1,000 simulations that we did for each of the conditions, for each of the 4 degrees of network correlations, and the orange lines in the plots are the averages across these 1,000 simulations. And what you can see here is that depending on the network correlations, the 2 state variables as B and as A stay longer time or more time at the red separatrix. So if you look at the orange lines, you will see that the more correlation we have, the longer the state variables are close to the red separatrix. Once a state variable moves away from the separatrix due to the configuration of the flow fields, the velocity towards an attractor state increases, and it becomes increasingly difficult for subsequent stimuli to pull the system back in the balanced state close to the separatrix. So as you can also see in the upper panel, there you see the distance of the state variables to the separatrix and the time. And we see for the red plot that stands for the high correlation in the network, the system stays much closer to the separatrix, to the balanced state that allows longer evidence accumulation, longer averaging of noise, and therefore the evidence gets more prominent and leads to the correct decision. So this is how we can use modeling and in particular tools from dynamical systems theory to understand the laws that link together structural connectivity, functional connectivity, and cognitive performance. So this was now for the healthy brain. And now I want to show you some examples how we use brain network modeling also to understand the diseased brain. So this is an avatar from a stroke patient. These data come from our colleagues from Irvine. They now moved to Dallas, Anna Solotkin. They have virtualized 16 stroke brains and inferred the model parameters from these individuals. And they have shown that the model parameters are predictive for the functional outcome for these 16 stroke patients after rehabilitation therapy. So on the right side in the diagram, you see one of the model parameters, K12. This is the coupling between the excitatory and inhibitory population. And on the y-axis, you see the Fugl-Meyer score. This is a clinical score that quantifies the motor performance of these patients. And there's a good correlation between the model-derived parameter and the outcome, the functional outcome for the patients. Here I show you another example of a simulation of the diseased brain. This is a brain with epilepsy. It's a 40-year-old woman from Marseille, also virtualized as intracranial electrodes. Unfortunately, this is again not animated here on your slide. So what we see is the onset of the seizure activity, and this is the intense black trace that then starts propagating across the network. It's reaching other regions and then terminates in the contralateral temporal lobe. So this seizure activity can in part of the patients be treated with drugs, but 30% of the patients are not responsive to these drugs, and therefore the only help for them is neurosurgical treatment. For the neurosurgeon, it is important to know where does the seizure start and how does it propagate through the brain. And so this is the starting region is called the epileptogenic zone, and then we have also the propagation zone. This is the relevant information for the neurosurgeon that classically is inferred by a team of clinical experts, human experts, from the data that are available for the patient. So here on the left side, you see such an estimation of the epileptogenic zone and the propagation zone in red, based on the clinical expert team. And in green, such a prediction has been made by the virtual brain. And the yellow area is the overlap between the predictions of the human expert team and the computer model. On the right side, you see now the outcome for, I think, 16 individual patients. Each dot represents one patient with epilepsy. The x-axis shows the Engel score, the clinical score after surgery, indicating the degree of improvement. And the y-axis shows the divergence between the predictions of the human expert team and the computer model. So the Engel score 3 indicates that there's no improvement for the individuals. 2 indicates that there is a decrease of the seizure frequency, and 1 indicates that the individuals were seizure-free after surgery. And what has been found was that the divergence, the difference between the predictions of the computer model and the human expert team was predictive for the outcome for the patient. So in other words, the model was able to predict the outcome of the patient. And now there's a clinical trial running in France that includes 400 patients. 200 of them will be treated with the addition of the information obtained from the virtual brain, and the other 200 gets a classical neurosurgical treatment. And at the end of the 4 years, it just started, we will know whether the inclusion of the predictions of the virtual brain improve the outcome for the patient. Here, this is a study from our colleagues in Belgium and at the University of Ghent, Daniel Marinazzo. They have virtualized the brains of patients with brain tumors. And they found, beneath other observations, that the model parameter J, the feedback inhibition parameter, is can differentiate between tumor tissue and non-tumor tissue, which could be also important information for the neurosurgeon in future. So here I show you a study with the virtual brain on dementia. Here data were used from the Sydney Memory and Aging cohort. Each of the dots again represents a patient, either healthy control, Alzheimer's disease, or mild cognitive impairment. And in the study it has been shown that the biophysical parameters inferred from the model are predictive for the cognitive scores. But even better, one now can attribute changes of individual parameters with a biologically plausible interpretation to the changes of the cognitive state. We recently also included PET data in our simulations. So in dementia patients or patients with Alzheimer's disease, there's an accumulation of amyloid beta, which can be measured with PET. And in vitro and in vivo studies, it has been shown that in the vicinity of beta amyloid plaques, the function of inhibitory interneurons is disturbed. So this is what you can see here. In the second column, in the presence of amyloid beta, the postsynaptic potentials, so inhibitory potentials of the inhibitory interneurons are decreased in amplitude, and the firing rates are also decreased. So now we can incorporate this information as a hypothesis in our models. So we use the information about the beta amyloid burden coming from the PET data of the individuals. Here we used data from the ADNI cohort. And we have formulated a cause and effect model, how the increased beta burden would affect the population dynamics of our individual nodes. So the model contains the fact that in the presence of beta amyloid, the activity of the inhibitory interneurons is disturbed, and this leads to disinhibition, and this leads to hyperexcitation. So this is our assumption that is now incorporated in the model. We use for population models, Janssen-Witt models that you can see here, that consist of 3 populations of neurons, indicated here by the 3 columns. The left one shows the excitatory interneurons. The middle column shows the pyramidal cells, and the right column shows the inhibitory interneurons. Now we modulate the impact of the inhibitory interneurons to the pyramidal cells as a function of the beta amyloid burden in each individual region. The beta amyloid we take from the PET data. When we do this, we see that just by including the beta burden coming from the PET data, the simulated EEG activity changes between the mild cognitive impairment and healthy control individuals and the Alzheimer's disease patients. So the red dotted line indicates that there's a increase of regions with frequencies around 4 Hz, which is the theta rhythm. So there's a slowing of the simulated EEG observed in our data just by including the beta burden and the mechanisms that I have explained. The slowing and the theta activity can be also observed in the empirical data. So this is now another tool, approach, method from dynamical system theory, bifurcation analysis, that nicely illustrates how we can make benefit of these tools for understanding possible mechanisms in the brain. So here in the 3 panels, we only change one parameter that is related to the coupling of the inhibitory interneurons to the pyramidal cells, it's tau. And just by changing the value of this parameter, independence of the beta burden that we derive from the PET data, we see how the repertoire of the system changes from a single limit cycle generating alpha activity in the top left panel to 2 limit cycles generating theta and alpha activity in the top right panel to just 1 limit cycle generating theta activity in the bottom left panel. And these are the corresponding time series. So on the top left panel, we see the alpha activity, and the top right panel, the mixture of alpha and theta activity, and in the bottom panel, the theta activity. So this is how a change of a parameter in the system changes the dynamic repertoire of our population model. It's also possible to simulate drug effects on the computer with this approach. So here we simulate the effect of memantine, which is an NMDA antagonist that is used in dementia, also in Alzheimer's disease. And we simulate the effect by blocking the NMDAergic transmission from the excitatory interneurons to the pyramidal cells. And as you can see in the right plot, the y-axis are the frequencies. We see the slowing of the simulated activity for the Alzheimer group, where we included the beta burden from the PET data. By just then blocking the transmission from the excitatory interneurons to the pyramidal cells, we can normalize the trace for the Alzheimer patients to the dotted line that is very similar to the behavior seen in mild cognitive impairment and healthy controls. The ultimate goal with this simulation approach is to identify biomarkers that better predict or classify different groups of dementia or in general diseases and ultimately also are able to predict disease trajectories. So this is a little bit older result. We have now a newer one that we just presented at the SFN conference in Chicago, where we provide proof of principle that by simulating features, by including imaging data, we can increase the accuracy of our predictions. So if we compare predictive power based on imaging data with predictive power based on simulated features, we can show that the predictive power is increased, and also the sensitivity and specificity in the area under the curve. So this approach is at the heart of a new project that I'm coordinating that just started, Virtual Brain Cloud, where we use the Virtual Brain software and multiple data sets coming from many of the 17 European partners that are part of the consortium. This project is funded with 15 million euros, and one goal is to get these data harmonized and processed in a standardized way in order to make benefit from this, this large amount of data that is available and distributed across different sites, and identify predictors. And with this, I would like to end my presentation. I hope you could understand it well enough, and I'm happy to take questions. I want to also mention actually that I want to thank my lab in Berlin, and particularly my colleagues from the Virtual Brain team, Randy McIntosh and Victor Giese, who you see here in the upper left corner.
All right, thank you, Dr. Ritter.